welcome to the another new video. Today's video will cover a bit new content as I'm going to talk from agricultural perspective. And in today's video, I'm going to cover about genetic engineering and crop improvement. Then, why do we need to improve crops? Well, first reason to improve crop is to have better production. The growing population rate and the current production from our existing agricultural system is not enough to feed this population. So, in a future, it might be a very big question like is our current or existing agricultural system would be enough, like would be producing enough to feed these peoples? So we should think to improve the production and productivity. So from the genetic engineering or biotechnology, we can improve our existing agricultural system to be more productive by improving crops for better production and productivity, so that we can secure food for future generation. And there are lot of people who are under the poverty line, so we have to think for themselves, for them as well. Mean we have to feed these peoples as well. So to maintain the food security and to maintain the food production and productivity level to feed a current and future population, we have to improve our crops. And another reason is for better adaptation. Our current globe and our agricultural system is facing a lot of challenges and problems such as insect pest, disease, weeds, and climate change. Insect pest, disease, and weed infestation is like havocly increasing. And climate change is a global concern where the global warming and greenhouse gas emission is taking in concern. So our agricultural system should have go through these type of challenges and problems. So in future, we should have our agricultural system to be adaptive for this kind of infestation and climate change and global warming, so that we can maintain our agricultural system even in the harsh situation, because agriculture is main source of or main way of living. So we have to improve our crop to be more productive or to be more adaptive, or both, based on the situation, condition, and our requirement. How genetic engineering improves our crops? Genetic engineering itself defines a manipulation process which either edit, either delete, or silence some of the plant genome, so that the beneficial genome we can either express, or the harmful or the unwanted genome maybe we can silence or deactivate them. So in this way we can either improve our existing plants, our crops, or we can improve completely new varieties by doing the manipulation of plant genome. And the new or improved plant varieties either would be resistant to the harsh situation or maybe high yielding. In this way we can improve our crops using genetic engineering. What are the genetic methods to improve our crops? Through breeding, through mutation, and through polyploidy method. Breeding in agriculture for plant help to exchange the traits between plants in order to produce desired characteristics, so that the breeded plant will have higher chance for yielding or higher chance for adaptation. There are different ways we do breeding using the genetic engineering in plant in agriculture is hybridization. Hybridization is the process of interbreeding between individuals of different species or genetically divergent individuals from the same species. It means it will breed between the genetically similar or dissimilar plants to produce hybrid, and the offspring produced by hybridization may be either fertile, partially fertile, or sterile, but it have or it the hybrid have higher yielding potential. And another one is tissue culture. We all know the meristem tissue, the apical meristem is free from disease, pest, and virus. So that we can take the meristem tissue from the apical meristem, and we can grow and we can develop healthy seedlings or saplings for future generation, so that the future generation of that plant will be free from virus, disease, and other pests, so that it would be adaptive. And another one is crossing. In science or in molecular genetics for agriculture for plant, crossing mean either back cross, full sheep cross, half sheep cross, or other different type of crossing. Crossing mean exchanging genetic materials between plants and species. In this way, we can either transfer the genetic material from the different species or different plants to the new plants, or we can also like delete, or we can also remove the undesirable or unwanted traits in existing plant, so that we oftenly do the crossing. And in some cases we can check the genetic purity of certain plants by crossing through the back rows. And crossing yeah, usually help to transfer or help to import the genetic material from the other species, and sometime it helps to like remove the unwanted or desirable traits. And another is mutation. Mutation is manipulation of genetic material, either through the gene editing or through the gene modification or through the gene addition, deletion, or silence. You know, in a plant, we all plants have different type of genome set, so we can edit this genome to add some desirable characteristics, or to delete some unwanted characteristics, or to deactivate the expression of certain unwanted or undesirable characteristics. There are different way to do this, either CRISPR, either Cas9 CRISPR, or zinc finger nucleases, or talents, or other gene editing tools, or any recombinant DNA technology which allow our plant to be mutated or having new kind of genetic material. In this way, we can improve our crop to be more productive or more adaptive. Another one is polyploidy method. This polyploidy is the presence in a cell or more than a single pair of each chromosome. And polyploidy can be result of a spontaneous multiplication of plant genetic material through the hybridization. And the polyploidy help to multiply the genetic material, and we can multiply the desirable genetic material or genome, so that we can have the more desirable traits on the plant. Or we can do the advancement of some genetic material by gene multiplication or polyploidy. Or we can do the gene improvement through the polyploidy, so that our desirable traits or desirable characteristics would be more advanced or more improved and highly expressed. But this method may not be always like beneficial. However, the hybrid developed through the polyploidy have the high hybrid vigor, so that it could be beneficial in plant for making it more productive. How genetic engineering works in crop improvement through the breeding, for hybridization, tissue culture, and crossing. How it works? It brings desirable property through any process, either hybridization or tissue culture or crossing. It brings desirable property or traits or characteristics to the plant. It brings disease-free seedlings from tissue culture and remove the bad properties or bad traits or undesirable traits. And how it works for mutation through the gene editing, modification, deletion, or silencing. It introduces the desirable genome by gene addition, and remove unwanted genome by deletion, and improve functional but not active genome by silencing. And we can give examples of different genetically modified organisms in agriculture, such as golden rice, bt cotton. And how it works for polyploidy. by multiplying, advancing, and improving genetic material in plant. It advanced the genome from functional aspects and multiply the desirable and functional genome so that our desirable characteristics or our wanted or the most desirable, I want to say desirable characteristics or trait would be multiplied in advanced and improved so that we will have that property. For example, if a plant is known for like high yielding, we can multiply that gene, high yielding gene, so that we will have the more production in next generation. Then, is there any example for successful application of genetic engineering in crop improvement? Well, yes, we have. First, I would like to go for the research studies. I mean, we have a lot of advancement in research studies, and scientists have developed a model plant for genetic engineering studies in plant, which is Arabidopsis thaliana. It is known for different name in different country and continent, but scientifically it is called as Arabidopsis thaliana, which is model plant. And we are like not we scientists and most of the other technicians are working with this plant. First, they do study in this plant to replicate the same like experiment with our other agricultural plants for transformation, for making GMOs and some other addition, deletion, and hybridization procedure. First, they do test with this plant and then they replicate with the other plant. So this is the like non-example that scientists have developed model plant for the plant genomic studies or plant genetic studies. However, we do have many other examples that we have achieved so far from the genetics or um, genetic engineering to improve our agricultural crops. So I do have some examples. First is Xanthomonas wilt resistant banana in Africa. This wilt like had damaged many banana orchard or banana field in Africa. And you know, Africa, like in Africa, banana is a staple food source. So introduction of the harp gene from the capsicum, scientists have like developed new variety of banana which is Xanthomonas wilt resistant banana. And another one is tearless onion. In New Zealand, this research is being in progress so far, and this research is being carried out through RNAi or gene silencing method. Another one is golden rice. I have given another lecture in my previous video, so you can go there and find what is golden rice. But I'm just going to introduce what is golden rice in a brief here. Golden rice is a new genetically improved or genetically modified rice variety which is rich in vitamin A, so that it helps to prevent vitamin A deficiency related problems in human, and gives carbohydrate source as well. So it's a good combination of food for people who have like rice as a main staple food source. So how it was success, like how it had got success, is through the introduction of beta carotene in rice variety. And another one is purple tomato, which prevent cancer, and it has developed in like Israel through the anthocyanin development gene. Another one is calcium-rich carrots. This carrot is useful to prevent calcium deficiency related problems such as osteoporosis, so that people will have the health benefit by consuming this carrot. And it like was got success through the introduction of CAX1 gene. And another one is healthy frying oil from soybean. It is like developed through the gene silencing, and this oil has low saturated fat, low trans and fats, and no hydrogenation required for this oil, and it is highly stable during baking. And another one is Arctic apple, which has no browning characteristics. You know, once we cut our apple used to get brown, but this Arctic apple, it has no browning characteristics, and it had developed through the silencing of polyphenol oxidase. And We have some like other achievements such as BT cotton and seedless watermelon, disease-free bananas, and like seedlings through the tissue culture. And I have already mentioned about the golden rice, art apple, and purple tomatoes. Thank you so much for keep watching my video till end. Please do not forget to like, share, and subscribe my channel, and do not forget to keep motivating me, keep supporting me. Thank you so much.